We wanted to understand protein loss through a dialyzer during the hemodialysis session. We know that dialysis, of course, means, first of all, to remove toxins from the blood. And if the filter pores are larger, more of these harmful middle-sized toxins can be cleared. That's the idea behind it, and that's the idea behind the so-called medium cutoff or MCO membranes for the dialyzers. But the question really is, do these larger pores also allow beneficial proteins to escape and to be removed, particularly also compared to standard high-flux dialysis membranes? And if so, how much more? So that's where our question started, and this is the research question that we answered with this study.
Instead of study real patients, we ran a lab experiment. We circulate a tank of human plasma through both a high flux and an MSO dialyzer at the same time. Then we use a very sensitive method called mass spectrometry to identify and semi-quantify all the proteins that came through into the ultrafiltrate. This gave us a direct side-by-side comparison of protein loss across the two membranes without the interference of patient's physiology. We found that the ultrafiltrate contains a wide variety of proteins. In total, we identified 244 proteins and could measure 113 of them precisely. On average, the MCO membranes allowed 8 to 9 times more protein to pass through compared with the high flux membranes. This means MCO membranes indeed leak more proteins, which raise important questions about the long-term impact on patients' health.
What's the significance of our findings? Now, when you think about it, MCO membranes, the medium cutoff membranes, they have larger pores than normal membranes, normal high flux membranes we use every day. And MCO membranes, like any dialysis membrane, they do not differentiate between good and bad substances that are removed. The removal of a molecule depends on its molecular weight, on its charge, on its shape or size. And these are the main determinants. Now, it happens that there are a number of beneficial molecules, molecules that are important for bodily functions, are also removed by MCO membranes. So what happens is that, for example, proteins that bind hormones are removed by MCO membranes. We found that specifically one protein that binds vitamin D is removed by MCO membranes to a larger extent compared to high flux membranes. We also learned that another very important molecule, haptoglobin, that plays an important role in the defense against oxidative stress, is also removed to a larger extent by MCO membranes compared to high flux membranes. And it will be really important to see in the future how this removal of beneficial molecules really affects the patient's health and well-being.
Our full article is available at the American Society of Artificial Internal Organs, or short ASAIO journal website. And if you have questions or ideas, we would be so happy to connect and talk more about how these findings could shape the future of dialysis care.